This is myosin, a thick filament. And this is actin, a thin filament. The two heads link the actin and myosin together during contraction. The ends of a myosin filament contain the heads, and there is a central bare area. The heads contain ATP binding sites. Active sites are blocked when the muscle is relaxed. We are now going to look at the structural components of the cell. This is the T-tubule. These are the synaptic vesicles containing acetylcholine. This is the mitochondria. This is the junctional folds of the sarcolemma at the motor end plate. This is part of a myofibril. This is the axonal terminal of a motor neuron. This is the synaptic cleft. This is acetylcholine esterase. During neuromuscular control, the axons of the nerve cells of the spinal cord branch and attach to each muscle fiber, forming a neuromuscular junction. An action potential passes down the nerve. The nerve releases Ca2+ that results in the release of acetylcholine. From here, acetylcholine binds with the sarcolemma. Next, we look at muscle fiber action potential. Acetylcholine binds with receptors and opens the Na+ channels. Na+ channels open and Na+ in. There is a decrease in the resting potential. From here, Na+ rushes in and the sarcolemma depolarizes. The regional depolarization spreads rapidly. The position patch in the membrane changes the adjacent patch of the membrane. Thus, depolarization spreads. The K+ channels open and the region repolarizes. Immediately after the action potential passes the membrane, permeability changes again. Na+ channels close and the K channels open. K+ rushes out of the cell and the cell repolarizes. In this diagram, Ca2+ is released from the sarcoplasmic reticulum. Ca2+ is stored in the sarcoplasmic reticulum. Depolarization releases the Ca2+. The Ca2+ clears the actin binding sites. In this diagram, we see that the neurotransmitter release diffuses across the synaptic cleft and attaches to the acetylcholine receptors on the sarcolemma. Action potential generated is propagated along the sarcolemma and down the T-tubules. Action potential triggers Ca2+, release from terminal cisternae of Sr. Calcium ions bind to troponin. Troponin changes shape, removing the blocking action of tropomyosin. Actin active sites exposed. Contraction. Myosin cross bridges alternately attach to actin and detach, pulling the actin filaments toward the center of the sarcomere. Release of energy by ATP hydrolysis powers the cycling process. Removal of a Ca2+ by active transport into the Sr after the action potential ends. Tropomyosin blockage restored, blocking actin active site. Contraction ends and muscle fiber relaxes. Sliding filament theory of contraction. During muscle contraction, the thin actin filaments slide over the thick myosin filament. When calcium is present, the blocked actin side of the actin clears. The myosin head is the red bulb you see in the picture. It is high energy configuration from ADP. Myosin cross bridge attaches to the actin myofilament. Working stroke, the myosin head pivots and bends as it pulls on the actin filament, sliding it toward the M line. As the new ATP attaches to the myosin head, the cross bridge detaches. The myosin head now has low energy configuration. As ATP is split into ADP and inorganic phosphate, cocking of the myosin head occurs. The end result is a shortening of the sarcomere. The distance between the Z discs shortens, the H zone disappears, the dark A band increases because the actin and the myosin overlap more, and the light I band shortens. Ca2+ is removed from the cytoplasm and tropomyosin blocks the actin site.